a lot of these statistical issues are fairly foundational in research design. How often do you see these things being violated? Is it like something you see every year?
Yes.
And is it the province of only? Is it also only the province of beginning private companies, or have you seen it also at larger concerns?
Well, you see it all the time, multiple times a year. You see more frequently in smaller companies because they don't necessarily have the statisticians and in-house reviews. At very large companies, I think most of these issues are caught. They're committed, but they're caught before they sort of make their way out of the labyrinth of big pharma. So very, very rarely will you see a press release from a large company with these kinds of issues. But you see it in press releases from small companies all the time. All the time. So, and it's not that people are necessarily trying to be less than forthcoming. It's just that these illusions are so compelling that it's hard for someone to not believe they're real. It's just the way that our brains are hardwired, that in every day, this is how we process information. So it's kind of like an instinct almost. Let's say you're playing basketball and somebody fakes you out. You get faked out even though you might be anticipating it. It's just the way your reflexes are. So these are almost reflexive.
If I'm a small company,
yes.
Will I get this type of research design help from? Should any statistical consultant be able to help me with this?
No, because there are a lot of statisticians who do statistics without understanding the fundamental underpinnings of statistics. So that's number one. You give them the data, they'll crunch it, and they'll give you the results. But they're equivalent of accountants, not financiers. So a good analogy is on accounting, you can give somebody a bunch of numbers and he can add that all up. But if you ask them, what does this mean, or how can we arrange the financing to do it that way, they won't be able to do it because they're used to just adding up the numbers. Only a minority of statisticians really understand the principles that underpin the statistical techniques they use. So that's number one. And number two, statisticians can't do this work unless they're provided with the right context by the clinician. So for example, one of the common mistakes is all statistical tests rely on certain assumptions. So for example, a normal curve. Distribution of values is normal. Most clinicians don't realize that. And furthermore, the distribution in both sample sets have to be similar. They can't be very different. Statisticians is not going to know that blood pressure has a non-normal distribution unless the clinician tells them. The problem is that a clinician may not know what they need to tell the statistician. So when the fundamental assumptions are violated, there's nothing a statistician can do if they don't know about it. So a really good statistician who has worked a lot in clinical research will know these, but you can't just ask any statistician.
You're in the midst of writing a series of books on clinical research.
Yes.
The conduct of clinical research.
Yes.
What motivated you to do that?
That book, the first book, grew out of an educational lecture series I developed when I was at Genentech to train new medical directors. Because we recruited pretty heavily from Stanford, UCSF, academic centers, very, very bright people, but who are new to industry. So I put the lectures together, and that eventually evolved into the book because I figured, well, I've done all this work already, so I might as well turn it into a book. And then I found out I liked it. And I also found that as you're doing it, you learn stuff. So that's why it led to the second book and third and so forth.
When is the last one due out?
Well, we have one. I have a book on global clinical trials that should be out in Q3, and I have a book on adaptive clinical trials that should be out about the same time.
I'd love to walk through it. How are we doing?
I'd like to start with an example of a sort of everyday intellectual optical illusion. So if you ask somebody, okay, you're in a foot race and you're running, you're running and you're in third place and you pass the guy in second place, what place are you in now? Most people say first, when in fact you're second, right?
Yep.
And that's just how the way our brain processes information, especially verbal information, right? Another example is, let's say you have pencil and eraser and they cost $1.10 put together. And the pencil is $1 more than the eraser. How much is the eraser? And most people say 10 cents, but in fact it's 5 cents. And some people like don't believe it and they have to work it out because they can't believe it's 5 cents. But that's, our brain has certain shortcuts that, or reflexes essentially. And 90% of the people, if you ask these questions, they'll answer it the wrong way. Now, it's worse when you're looking at aggregate data instead of everyday data, because everyday things is what our brains are hardwired for, sort of anecdotal stuff. So when you have massive amounts of statistical information and you're trying to process it, that's not what our brain was designed to do. And primate brains are not hardwired for that, because it's only recently that we've had access to that kind of access. So the first is regression to the mean, which most people understand, but still it's very common. So let's say you have a drug and you just think of depression and it doesn't work. It fails to meet the primary endpoint. There's no difference between drug and no drug. But your preclinical guy comes and says, oh, of course it didn't work. I told those guys not to design it that way. All the animal data suggests that it's only going to work if the disease is really severe. So you take the data and you say, okay, let's just look at the severe patients, and you realize, oh my God, look at this. You get to a 0.001 if you just look at the severe patients. So you decide to go to phase 3 and it doesn't. And of course, the problem with that is depression, like many other diseases, you have waxing, right? So if your sort of median is, 5, some days you're going to be 8, some days you're going to be 2. So if you take a group of patients and you just take the most severe, even if you do nothing, 8's going to become 5 on average, right? So the things you have to do is, don't look at the treated patients only. You got to look at the placebo patients, because if it's regression to the mean, what you'll find is that even if they get placebo, the most severe patients get better and the least severe patients get worse, just because of regression to the mean, right? So whenever you're segregating by subgroup, especially if it's by disease severity, you have to be very, very careful. And this further translates into one of the reasons why so many phase 2s fail in phase 3, because just by random chance, you're going to have some distribution of results in phase 2, some of which will be positive on a spurious basis. So if you only advance those, then you're likely that, ultimately you would expect phase 3 rate to be lower than phase 2, even if everything was perfectly designed. So that's the first illusion. And that most people who've had training in even elementary statistics know about it, but it's very easy to fall into that trap. The second thing is survivor bias, which is, let's say you have a drug, you're developing it to treat MI patients. And when people have MI, they can die or they can have CHF. Now, with modern therapies, only a few people die, so the power is very low unless you have very, very large studies, right? When the mortality rate is 3%, then you need thousands of patients. So what's commonly done in those situations is you get a composite endpoint. So you say, okay, well, what else, is a bad sequelae, and you add it together. So you say death and CHF, right? In other instances, you say death is too uncommon. Let's just use CHF. Because, when you have composite endpoint, there are some issues around that too. So let's say you're developing a drug, you decide, okay, death, it's only 3%. To show a difference in that, that's difficult, but, CHF, maybe it's 15, 20%, so we'll use that as an endpoint. And you do a study, and in the treated group, patients with congestive heart failure goes up. Like, okay, now the drug must not work. But the problem is this. If your drug prevents people from dying, often they won't die but end up with CHF. So your drug that's preventing mortality can increase the number of patients with CHF, because those patients who would have died, they have less heart damage, so they survive, but they have CHF. So you get survivor bias. You have to be very careful when you're looking at these endpoints, because you have to ask, okay, if our drug really works, is it going to result in a paradoxical result? So, and then the other illusion is, this is actually less common. So let's say you develop a seizure drug, and seizure drugs often have a very narrow therapeutic index, and you want to see whether the drug has better sort of dose response curve, because if you have a gradual dose response curve, that means your therapeutic window is wider, right? So you do it, and the therapeutic window looks better. But the problem is that when you're looking at a dose response curve, it's an aggregate number, right? So the dose response curve in individual patient may be, quite steep, but if the variability from patient to patient is greater, it smoothes out the curve. And once again, this is one of the issues that you run into whenever you're aggregating data, is that when you aggregate data, it kind of looks like individual data, but it's not individual data. And usually some reasonable correlation between individual and aggregate data, but not always. And then the next thing is also illusion 4 is something that a lot of people learn about, but it's sometimes hard to apply in life. And when you have a drug that reduces mortality by 90%, or reduces mortality by 9%, or reduces mortality from 10% to 1%, or 90 to 99%, those are all the same, because going from 1% to 10% is actually the same as going from 90 to 99, because let's say you increase survival from 90 to 99%, you're decreasing mortality from 10 to 1%, right? And a good example is actually like weed killers. You look on the package and one bag says kills 99% of the weeds, and another bag says it kills 99.9% of the weeds. So, oh, this sounds the same. But you realize one that works 99%, you end up with 10 times as many weeds, right? And once again, our brain is not trained for a program to think in what are called odds ratios. There's some data suggesting that when babies are born, they think in odds ratios, but as we grow, we kind of get trained out of that to thinking, straight percentages. So when you're looking at study results, it's important to realize that you can flip it around, and that's very important. People who do drug development in virology, like hepatitis C or HIV, think much more this way, because they talk about log reduction. But that's the exception. And then p-values, I think you're familiar with. Just because a p-value is higher or lower doesn't mean there's more of a difference, right? Because if your study's larger, you just get a smaller p-value, even with the exact same amount of benefit. So one study, there's a 10% benefit, but it has 30 patients, versus another study, there's a 10% benefit, but there are 3,000 patients. So the p-value is going to be much, much bigger in the second study than in the first study. P-value just tells you, given the set of patients, how certain can you be about the data. It doesn't tell you what the magnitude of the effect is. And then a few other things. One is, p-values are fine for efficacy. P0.05 fine for efficacy, but certainly, as we learned from Vioxx, you can't apply that cutoff to safety, because no one's going to go out and say, oh, there's only 80% chance that this drug is causing harm, so we won't worry about it. It's sort of like saying, oh, I'm not going to lock the front door to my house because I'm only 90% sure that it'll get robbed tonight. So until I'm 95% sure, I'm not going to lock the door, right? And that's sometimes how people approach illusions. Post-hoc analysis, and I think this is something that many people are familiar with because people have been burned so much, which is you do these analysis, post-hoc analysis, enough time, sooner or later, you'll get some positive p-value. Statisticians like to say if you torture the data long enough, then sooner or later it's going to confess, even if it's false confession. So you only get one bite at the apple.